warm good morning to one and all present over here. This is Ansika Srivastava of MSc second semester microbiology going to deliver my presentation on the topic lac operon. These are my contents. Now, introduction. Operon is an operating unit which can be defined as a cluster of genes located together on the chromosome and transcribed together. It is a group of closely linked structural genes and some associated genes which regulates in the metabolic activities. The model of operon is first described by Francois Jacob and Jacques Monod in 1961 at Pasteur Institute of Paris. Now let us discuss about the types of operon model. So there are two types of operon model. Number one is inducible operon and number two is repressible operon. Number one, inducible operon. They include genes that encode for enzymes that take part in metabolic pathways. The expression of the gene is controlled by the substrate, and their example is lac operon, which we are going to study further. And number two is repressible operon. They include genes that encode for enzymes that take part in biosynthetic pathway. The expression of the genes is controlled by the end product of the pathway. For example, tripoperon. Now lac operon. E. coli can use lactose as a carbon source. Lactose, as we know that lactose is a disaccharide made up of glucose and galactose. The lactoperon codes for the enzyme involved in the catabolism of lactose. It is the inducible operon since the presence of lactose induces the operon to switch on. Now, what are the elements and their purpose in the lactoperon? So number one is operator, that is lac-O, and it acts as a binding site for repressor. Number two is promoter, that is lac-P, that acts as a binding site for RNA polymerase. Now third is repressor. Repressor, that gene encoding the lac repressor proteins bind to the DNA at the operator and blocks binding of RNA polymerase at the promoter. Now the last one is inducer, that is lac-I. Lac-I control production of repressor protein. Now this diagram shows the structure of lac operon, and here all the essential component of operon is there, that is inducer, the operator region, the promoter region, and all the three structural genes. Now this table shows about the lac Z, that is the structural genes that codes for enzyme beta galactosidase, and their main function is to break down lactose into glucose and galactose. Lac-Y, that is galactose permease. This protein found in the E. coli cytoplasmic membrane that actively transports lactose into cells. Lac-A, that codes for the enzyme thiogalactosidase transacetylase, and the main function of this enzyme is not known yet. It is coded for gene lac-A. Now let us study the functioning of lac operon. There are two conditions based on the presence and absence of lactose. Number one, in the absence of lactose, the regulatory gene produces a repressor protein which binds to the operator site and prevent the transcription. As a result, the structural genes do not form mRNA and proteins are not synthesized. Number two, in the presence of lactose, when lactose, that is our inducer, introduced into the medium, binds to the repressor and repressor fails to bind to the operator. Therefore, the operator remains free and induces the RNA polymerase to bind to the initiation site on promoter, which results in the synthesis of lac mRNA. This mRNA codes for three different enzymes necessary for lactose catabolism. Now, functioning of lac operon. Here in this diagram, it is shown that when lactose is absent, then the lac repressor binds tightly to the operator. It gets in RNA polymerase's way, preventing the transcription. And in the second diagram, we can see that in the presence of lactose, allolactase, that is rearranged lactose, binds to the lac repressor and makes it let go of the operator. RNA polymerase can now transcribe the operon. Now, in this table, there are different scenarios observed regarding the presence and absence of glucose and lactose. In the first one, where the both glucose and lactose are present, when both are present, RNA polymerase can sit on the promoter site, but it is unstable and it keeps falling off. So there is no transcription takes place. In the second one, where glucose is present and lactose is absent, then there is no transcription occurs, and that's because the lac repressor remains bound to the operator and prevent transcription by the RNA polymerase. In the third one, where both glucose and lactose are absent, then the protein bound the activator protein bound to the DNA and repressor bound to the operator site, and RNA polymerase blocked by the repressor, and no transcription takes place. And in the fourth one, where glucose is absent and lactose is present, another protein is needed, that is activated protein. This stabilizes RNA polymerase. The activated protein only works when glucose is absent. In this way, E. coli only makes enzymes to metabolize other sugars in the absence of glucose. Here in this figure, as we know, lactose is present, then the repressor protein binds the operator, hence block the RNA polymerase to bind to the operator site, and hence transcription is not takes place. In the second diagram, lactose is present, then the operator site is free, and then RNA polymerase can bind to the operator site, and hence the transcription goes on. And in the third one, where both lactose and glucose is present, then repressor protein removed and RNA polymerase are bound to the promoter site, and hence transcription can occur smoothly. In the fourth one, the lactose is present and glucose is absent, then activator protein steadily the activator protein only works when glucose is absent. So in this way, E. coli only makes enzymes to metabolize other sugars in the absence of glucose. Now, the two types of control. The lac operon is under two types of control, that is positive control and negative control. The first one is positive control. When glucose is available, genes that participate in the metabolism of other sugars are repressed, and the phenomena is known as catabolite repression. It is a type of positive control in the lac operon. A catabolite activated protein, that is CAP, bind near the site of promoter and stimulates the binding of RNA polymerase. Now the negative control. The lac repressor binds to the operator. The DNA sequence covered by the repressor overlap the DNA sequence recognized by the RNA polymerase. Therefore, when the repressor binds to the operator, the RNA polymerase can't bind to the promoter and transcription cannot occur. The lac operon is said to be under the negative control. Here the diagram showing the positive and negative regulation. In the first diagram, where the operator activator protein binds to the operator and hence transcription is going on, whereas in the negative regulation, the repressor protein binds to the operator site and hence there is no transcription going on. Thank you so much.